Pranams to our Secretary Maharaj. We'll start today's session. I welcome the speaker as well as the distinguished members of the audiences for coming to our sessions today, the Vivekananda Science Circle sessions. You know the title and all those things. I won't want to repeat because you know about that thing. Just a few words about the speaker and her background. And so I will do that, and I won't take much time actually. Yes. Dr. Malancha Ta, the speaker. Let me see. He's a distinct. She's already an accomplished with. She's all. I'm sorry that I'm getting. I wrote it in handwriting. My handwriting I cannot read. And Dr. Ta is already an accomplished scientist with great potentials to make many more contributions to the advancement of bioscience and biotechnology, particularly in the relatively new and exciting field of stem cell research. Malancha received her bachelor degree in bioscience from Delhi University, master's in biotechnology from JNU, Jawaharlal Nehru University, and PhD in life sciences from the National Institute of Immunology in New Delhi. She did her postdoctoral work at NIH Bethesda. NIH is National Institute of Health, central funding agency, and doing all kinds of work in health-related field in the USA, by US government. It's in Bethesda, USA, then moved back to India in Bangalore as a principal scientist of a private company named Stempeutics Research Private Limited, and adjunct professor at Manipal Institute of Regenerative Medicine. In 2011, she received Young Investigator Award at the 4th International Conference in India on Stem Cells and Cancer. So this is a brief introduction. So she's quite accomplished, and though she's very young, so I'll invite her on the stage actually. Thank you. On behalf of the Institute, we just, as a token of appreciation for the time that's spent by our speaker and giving the talk, we like to give her, this set of books actually from the Institute. Thank you.
Good evening, everyone, and thank you so much, Dr. Lahiri, for the kind introduction and the words. And so I'm Malancha from IISER, Kolkata, and so our campus is in Mohanpur, and so this is I'm totally grateful to the Vivekananda Science Circle for giving me this opportunity and inviting me to this for this talk, and to allow me to share my experiences with stem cells over these many years. And also it's a complete honor to be here because it's, though not directly but indirectly, I have been associated with the Ramakrishna Mission institutions. My father, many uncles, cousins, and now my husband, they all have studied or been associated with the Ramakrishna Mission at some point or the other. And as a child, I also have come here to this very much, the same place, Gold Park Ramakrishna Mission, and stayed at the guest house. And all that I remember, the place looked so beautiful and nice, and I loved the continental chicken at dinner and the mangoes. So that's all I remember very distinctly. So it's a complete honor and a very different feel today when I stand here to share about my work. So with that, I'll move on to the work part which I'm going to share today. So in general, when we hear the word stem cells, so as a general population, when we hear it in the newspaper or when we hear it in the news articles or anywhere, so it's like, I believe the most common or the most first thought that comes to our mind being that it is something which is used to treat patients or it is used in the clinics. So something, and that is kind of very, very right. And why the stem cells have become so popular as in the clinics or for its clinical applications, its therapeutic applications, which is benefits which it offers, being the fact that stem cell means it is a very early cell, so that is how we define, and it's a very early cell which can differentiate and give rise to different kinds of cells, different kinds of matured cells. So to simplify this, for instance, if a person has had a cerebral stroke, or a person has had either say some sort of an ischemia disease or a myocardial infarction, so other than the therapies or the medicines, this provides an alternate way of medicine which can provide relief in the way that the stem cells can be made to become other cells, the functional cells, in the lab or in a clinic, and then that can be transplanted into the patient. So say for instance a stroke patient who has suffered a huge amount of neuronal cell death and is not able to function or is not able to carry out with his or her regular functions, in case functional neurons from somewhere is put into this patient, then the person is again able to, the person would revive and would be able to, the way we say, hypothetically or principally, will be able to again function and work. So that is where the use of stem cells come in. So stem cell is a very early kind of a cell which can be used to make, what's the point of working? Which can be used to make, say for instance, nerve cells, liver cells, platelets, red blood cells, etc. So the blood cells and all, or in all these different kinds of cells, it can be used to make in the lab. So, and also, again, when we very simply define, stem cells are cells which can self-renew. When we say self-renew, it means the cells can divide. So in our body, generally, most of the cells can divide. So the cells can divide and they can repopulate or make more of themselves. At the same time, the cells can also differentiate. The term used scientifically is differentiate. That means a stem cell can also make a liver cell or a neuronal cell or a heart cell, pancreatic cell, etc. These stem cells can make. So if you think in a lab, if you're able to take stem cells and make, say, liver cells or neuronal cells or a heart cells, and the same cells can be put into a patient who has suffered some sort of a loss in those cells, then the person is able to again function properly. So that is where the biggest, largest benefit and the huge promise that the stem cell field offers. So is the slide moving forward with this? That's okay. I can do it from here. That's okay. Point it to the key battery loans. So the promise of stem cell research, for instance, you take, if you take like cells in a dish, And so we can try to identify different drug targets. Example, if a new drug has been made by a pharmaceutical company, and before the drug is launched, it has to be tested. So we cannot simply catch human beings from the roads and ask them to try out a new drug. So rather, if you take, for instance, if it's a drug for the heart cells, so you can make heart cells in a dish in the laboratory, and you can test the drug efficacy or how toxic is the drug or what sort of side effects this drug could be having. So all that you could be studying in a dish of cells in the lab. And those heart cells can be made from stem cells. So hence this stem cells can be used to identify drug targets. Toxicity testing, whether the drug is really toxic or not. If you take the drug, how many healthy cells are dying, or are the cells at all recovering? All these studies can be done. Similarly, to understand cell differentiation or during the developmental stages. So when an embryo is developing in the womb of a woman, so that time there could be many developmental disorders which can happen. So there are many inborn errors of metabolism which can be there. So again, to understand those, study those, we cannot really take out the embryo from a body, but rather we can try to study many of these things in a dish of cells and take the stem cells, make them to differentiate, and at these different stages of differentiation, we can try to understand the effect of a certain gene or how these developmental pathways are. Similarly, some of the things is also that the stem cell conditioned medium, so because these have a lot of regenerative properties, is often used even in the cosmetic, for different kind of cosmetic products also, where there is a renewal of the skin, the renewal of the cells on different parts of the body. So hence that can be used for, even in this cosmetic industry also, the cells and the conditioned medium have found use. So just to show you what broad variety of kind of uses can be there with the stem cells. So now, where are the stem cells found? So I've been telling you about stem cells, so where do we find them? So there are broadly two categories of stem cells. The embryonic stem cells, which are called also the basic thumb rule cells, or like these are the basic ones. The gold standard stem cells are the embryonic stem cells. And then there are the adult stem cells. And the term, as they say, embryonic stem cell means you derive it from an embryo. So an embryo is a developing embryo, which is from at that stage, at a 5-day-old stage embryo, which is called a blastocyst. You can make the stem cells, and that is an embryonic stem cell line. And also in each one of us, in totally fully formed adults, starting from a fetal stage through the neonatal stages, the different stages of as a person reaches adulthood until late age, we all have stem cells inside our body. So in the different tissues and organs, we have pockets of stem cells, and those are generally sitting quietly. Only when there is a damage in a tissue or when there is a problem, the cells come to life, the stem cells come, they get into action, they realize that, okay, now I need to make more cells and get the body cured. So this is what the main function of the adult stem cells are. So here if we see, so these are the embryonic stem cells is from a developing stage. So this is, for instance, a zygote, a single cell after the sperm and the ovum, after a sperm fertilizes the ovum, the zygote stage. When it goes into multiplication, there are certain divisions happen. In a blastocyst stage of the embryo is something like this. This is before the implantation into the uterus. And here then you have the tissue and the other kind being the tissue and the adult stem cells, which is found from a newborn baby in the fetus and into fully formed adults. So these are the two main kinds of stem cells. So coming to the embryonic stem cells, this was first derived, mouse embryonic stem cells were first reported in 1981. And 17 years later, in 1998, human embryonic stem cells were first reported by Thomson and his colleagues. So this was a landmark year in 1998 when for the first time human embryonic stem cells were reported. As you can see, it is not too many years, it's a kind of a new concept still. So hence there is still a huge amount of research which is going on into embryonic stem cells or in the field of stem cells in general. So here in case of an embryonic stem cells, because I told you it comes from the blastocyst stage of the embryo. Now where will you get this embryo from? So ideally speaking, an embryo would happen inside a woman's body after she has conceived. But then that is not where you can, it is not ethically correct. When we do science, we also have to keep in mind, especially in bioscience, what is ethically right and what is not. So the ethics do not allow us to take out an embryo from a woman's body. So it cannot be used like that. Rather, we get the embryo from the IVF clinics. So these are the in vitro fertilization clinics or in vitro fertility clinics. So where couples go in case they are not having children or there are certain problems. And that is where they make the sperm and the egg would be fused. And you can get the embryos in a dish or in the lab. And the bestest embryo is put back into the couple, because into the women who have come to conceive here or to have a child. And the rest of the embryos, the extra embryos, can be donated to a stem cell lab. So when these embryos are donated to a stem cell lab, that is where a stem cell biologist or a researcher can get embryonic stem cell lines from. So here if you see, so this is basically the cultured, the blastocyst stage of the embryo. This is done in vitro. When I say in vitro, it means outside the body. In vivo means inside the body. In vitro means outside. In a lab, when you do, in a culture, in a dish, when you are doing these experiments. And this is the inner cell mass. When you take this inner cell mass, grow them in a dish, you get this embryonic stem cell lines. And those now can be differentiated to neurons, cardiomyocytes, pancreatic cells, whatever you wish to. So this is how this stem cells are got, embryonic stem cells. So this is a cartoon of a typical blastocyst stage embryo. Inner cell mass, this is called. And this is what gives rise, these green cells here you see, this is what gives rise to the embryonic stem cells. So this one is taken out in a lab and you take it out and do the culture, it becomes an embryonic stem cell line. So this is again the same thing. So this is an in vitro fertilization day 0. By day 5, it is the blastocyst stage of the embryo. From here, this blue one is the blastocyst, you have the inner cell mass. So that if you take out, grow in a culture, in a dish, in the lab. So this is what a typical dish looks like in a biology lab. And these are the cells which are called this would be giving rise to the embryonic stem cell lines. So it is actually speaking, it is not as easy as I show it to you here. It's also very, it's technologically also, it's quite difficult to get these lines, because once you, as you can understand, a 5-day-old blastocyst is extremely small. So you have to do this whole procedure under a microscope. So I'll show you, I'll tell sometime later, towards maybe the last slide. So I at one point was working with Reliance Life Sciences in Bombay. So that is where we had this in vitro fertility clinic. We used to get the embryos. And under the microscope, I have seen with what difficulty it has to be a very vibration-free stage microscope. The room should be vibration-free, etc. How when you're looking under the microscope, needles going from both sides, and these cells are, this blue cells, this is not blue in reality, but this is what is just represented here as a cartoon. So these cells are taken out and started as an embryonic stem cell culture. So these things are like quite, you need people who are really trained in it, and anyone, most of the time, most of the people will not be able to do, we won't even have steady hands to do this. And then the stem cells can be used to make different kinds of neurons, blood cell, etc. So why are these cells, what are the advantages of these cells, or what are the good properties of these cells? They can self-renew, that means you can go on and on dividing, expanding them. You start with little bit of cells, you can make lot of cells, because when you're going to put into patient, you would need huge amount of cells to be put into patient. So the cells you would have to take, because otherwise it's not going to work. So we generally try out these studies first in mice in the lab, then in human patients, etc. Then they have the multilineage differentiation capacity. The cells can be made into neurons, cardiomyocytes, in all kinds of cells. So that is what is one of the characteristic properties of embryonic stem cells. clonogenicity, which means from one cell you can make a group of cells. Because it goes on dividing, it can form a clone of cells. Again, clone is a word we hear normally. Often we joke about somebody, oh, this one is a clone of that person. That means a person is behaving very identical to this person. So a clone of cells mean when they are all identical to each other, a group of cells. Then there is a normal karyotype. So the karyotype is again something which, if you're lucky and don't have any genetic disorder, maybe we need not understand this word, but otherwise it's again something which you would have these days being talked about by regular doctors, etc. Because we often say, if a person, say for instance in late age pregnancies, or when a person is going to have a child with some certain difficulties, with like if both the parents have certain genetic diseases or something, so you want to test the chromosomal profile of the baby. So that is what is done in a genetic department in a hospital. Most of the hospitals these days have a genetic department. Karyotyping means to understand if the chromosomes all look normal and fine or not. So if the chromosomes have a problem, you get, example, something like a Down syndrome. Again, most of us understand what is Down syndrome, and this is a XXX example, if certain chromosomes have a problem in them. So that is what the cells would, even if they divide a lot, they still maintain normal karyotypes. Chromosomes all still continue to be normal and fine. Then extensive proliferation, they can proliferate for a long time. And also when we are doing experiments in the lab, we do not want to continuously plan and work and without any sleep or food or outing for us. So we have lot of extra cells, for instance. So the extra cells, we should be able to cryopreserve. Cryopreserve means you take them and put them into liquid nitrogen, which is at minus 196 degrees Celsius. You freeze these cells, and later some point you can again take out these cells and they will come to life and they will again continue to grow. So these are, you might have seen in some of the sci-fi movies also, where a human is frozen down fully, and after 60 years the person comes to life and is at that same age where he was frozen down. So that's like, might be a bit too far-fetched, but this is what a regular cryopreservation means of cells in the lab. So these are some of the properties which all the embryonic stem cells should be able to undergo. So these are some of the, this is like kind of a little bit of a, it's not something which we need to really understand. These are certain markers. So in biology lab, we often identify cells by the markers that they express. So example, if you have an insulin producing cell, a pancreatic cell. So the insulin producing cell, the beta cell of the pancreas makes insulin. So insulin is a marker for it. So in order to understand if your cells are fine or not, pancreatic beta cells, you would look at the expression of the insulin. Similarly, embryonic stem cells express certain markers like this. And they also express something called telomerase activity. And they can be again differentiated to, like I said, all kinds of cells they should be able to differentiate to. So this is how we typically, this is actually a real picture, this is not a cartoon. So when embryonic stem cells you are differentiating them through, say, heart cells or neuronal cells, you would go through a stage like this, which is called an embryoid body in the culture dish. So an embryoid body is something which is like, it looks like that. It is basically something like an embryo forming in the dish outside anyone's body. An embryo is forming. So it's like a tiny, very first stage embryo or a baby which is starting to form. It's like that. This is what it looks like. In the culture dish you would be able to see. So there's an interesting incident that had once happened to me. So I was working at Reliance that time. I had just got introduced to working with embryonic stem cells. And I had a dish of cells which was like this. And next day was a holiday or something. So I had worked till late. And I had forgotten about one single dish in the incubator. So incubator is like where we keep our cells and they are growing. It is like a warm, the way we say incubator, where a newborn baby many times has to be put because if the baby is not developing well, something like that is an incubator in a lab. So this kind of cells, so I had forgotten about one dish, I had missed to look at it. I took the local train, went back home in Bombay, and next morning when it was a holiday, after one day when I came back, So my sir in the lab asked me that, what did you do to this? I said, where did this come from? He said, it was in the incubator. So I said, oh, I'm so sorry, I must have forgotten about it. He said, do you know what have you done? So I was really getting worried by now, because I thought he was going to be a little upset, because I'd totally forgotten about this dish and these were important cultures. So he connected immediately the microscope to the computer, and he showed to me there was something like a beating happening. So this is exactly like a heart beating that had started to happen. This is a spontaneous differentiation, and the cells had undergone a cardiomyocyte-like differentiation and had started to form a heart-beating cell. And this is the capacity of embryonic stem cells. They can spontaneously do this. Spontaneously means without you having to do anything. It's like the way I had forgotten about it, and when I came, I saw this had formed. So he was actually not angry or upset. He was extremely thrilled and happy. So that is how that story ended. We knew how to make cardiomyocytes, at least. You put the cells and you forget about it for some time. So spontaneously also the cells would make. So this is just to show basically some of the components that you need to add in an embryonic stem cell culture. Each of these are different reagents that we add in the lab. It's not important to understand them here. And also to say that each of these components are extremely expensive. So it's a huge amount of expense, a huge budget we need to do embryonic stem cell culture alongside the expertise, etc. So this is again shows, just to show you what sort of a complicated protocol it is to make pancreatic cells, hormone expressing endocrine pancreatic cells starting from an embryonic stem cell. So these are different, you see all these yellow components are different chemicals or reagents that we have to add. Stepwise, not one thing can be forgotten or added earlier or later, because they follow a certain developmental pathway. And at each point, we look for these different genes. We all understand genes and markers. These are all the different genes you have to look for at each stage by doing PCR, protein, etc., looking at the protein, mRNA, RNA level, etc., to see if all this is expressed or not. So it's a huge, back-breaking, requires tremendous amount of patience, and at the end of it you might not even get those cells, because something or the other could have gone wrong. So that is the best part of it. So you need a huge amount of boost. You have to keep giving yourself the right kick all the time to say that it can be done and next time it would happen. So I think the greatest lesson we learn in basic research is not to lose hope and keep being holding on to dear hope all the time that something is going to come out of all this. So this is basically a typical steps before embryonic stem cells go till human trials. So embryonic stem cells have been tried. One of the biggest US companies, Geron, has been trying embryonic stem cell trials. They have been trying for many years now. Taking this, they have faced a lot of hurdles in the initial stages. So taking embryonic stem cells, making neuronal cells, and putting into the brains of people to see whether the brain activity becomes better. So this is this company called Geron. They were first ones to try this. So if you take human embryonic stem cells, first you have to ensure if you are making neurons, it is pure neuron population. It should not make heart cells, liver cells, pancreatic cells, and everything else there. It should be pure neuronal population. Only then it can be put into a patient's head. Just imagine if you're taking a close one, somebody of your family who has had a stroke or a heart attack or whatever, and you are taking for a stem cell transplantation, you do not want the person to get a mixed population of something you don't understand. You definitely want the person to get only neuronal cells if it's a cerebral stroke or whatever it is. So something like that. So it has to be first established to see that it is a pure cell culture, pure cell neuronals or whatever you have made. Test the physiological function. So it is not good enough to make only neurons, because inside the patient's brain, the neurons should work also. They should form the right electrical connections and all the stimulation should happen. So other than that, otherwise what is the use of making any neuron which doesn't function and putting into a person's brain? So the neurons should be, I'm just giving neuron because as an example, it could be anything else also. So these should be functional. Third is, like, if you are really serious about this work and taking it to the trials or to the clinic, the cells should be efficacious, that is, efficacy should be tested. How good are the cells, how much cells are getting differentiated, etc. you have to check in mice model. That means in the mouse, you have to make mouse or rats. You make a similar cerebral stroke kind of a model. Put the stem cells, the stem cell converted to neurons, put those cells into the mouse brain and see if the mouse now functions properly. If it does, next step would be in the non-human primate models. That is those huge monkeys or the chimpanzees kind. So you have these primates, again because they are closer to us. So in their brain also you create these problems, put the stem cell converted to neurons into the brain and see whether that is curing them also. Finally, you would be trying it in human. So before the human, all these steps are important. Then you show the demonstrate safety. It should not happen that it is very good, working fine, but moment it is put inside the brain, the animal is dying. So it has to be safe also. Then again you have to find out what dosage is correct. If the amount of cells that you're putting is enough, sufficient, it's not going to cause any other problem into the animal model or into the human that you're putting. And then to test methods to prevent rejection. So we all have again heard about something called, we all know about immune rejection, right? You know that you cannot take somebody's kidney and put into someone. We always know it has to be matched. Tissue matching has to happen. Otherwise it gets rejected. So or else you have to put heavy dose of immunosuppressive drugs. So whichever way, you have to find out what is right for this treatment, because these cells can get rejected also. So it won't, so the embryonic stem cells again, if you have taken and made like say neuronal cells or whatever from someone's embryonic stage cells, it could get rejected in our body also. So hence the rejection also has to be kept in mind. So after all these things are being fulfilled, finally we reach the human trials. Then also you don't know whether it would, how successful it would be. So for instance, just with respect to Geron, when they tried this neuronal cells into brains of patients, and they later found that there were small gliomas or like tumor kind of things forming in the brain. So then they had to immediately stop that trial. So these are the different kinds of hurdles people could face all the time. So now if we step out into the other kind, because embryonic stem cells have certain disadvantages. One biggest disadvantage being there are a lot of ethical questions about it. There are a lot of religions which believe that you are destroying a life. So if you take an embryo, it is basically killing a life. Even if these are extra embryos from an IVF clinic, still it was a potential life. And you're killing it for your research use. So many countries, this is not allowed. Embryonic stem cell, developing embryonic stem cell lines is banned in multiple countries all around the world. So that is one of the biggest hurdles of working with embryonic stem cell. Plus you need an IVF clinic and all those things. These are like a lot of hurdles on the way. A much easier source of stem cell is the adult stem cell, because these are the cells which can give rise to, you can get the cells, derive the cells from different kinds of tissues inside our own body. And also many of these tissues are discarded tissue. Discarded means they are anyway thrown into the dustbin. So these are tissues which we do not need. So for instance, in an example in case, like say a person who is really obese and goes to a plastic surgeon or a cosmetic surgeon for medical reasons, if the doctor has said that you have to reduce weight because the person is hugely obese or fat, and that is a concern, health concern, the fat tissue is taken out and thrown in the dustbin. Now if you take those fat tissue, that is a very rich source of adult stem cells. So the adult stem cell is much easier to get. Similarly, bone marrow, from the marrow of the bone you can get adult stem cells. So here these are some of the different kinds of tissues. This is for instance the adipose tissue, fat tissue, which is thrown out by a cosmetic surgeon or a plastic surgeon. This is for instance a bone marrow, so which is from the marrow of the bone, the femur bone, the longest bone. So you take out the bone marrow from there, the blood. And then there are a lot of birth associated tissues. So when a baby is born, you have the umbilical cord. That is cut and it is thrown away. So you can take that umbilical cord and you can get a source of adult stem cell. That's a very rich source of stem cells. In fact, in my lab, we work with the umbilical cord stem cells. Other than that also, there are multiple other tissues from where you can get the adult mesenchymal stem cells. So these are called the mesenchymal stem cells. So example, you can have the deciduous tooth, like when a child's tooth falls off, that is a deciduous tooth. So the tooth follicle also has stem cells. Our hair follicle has stem cells. If we pull out a hair, the follicle has stem cells. These are all adult stem cells. So they are present everywhere. Alongside the placenta. So as the child is born, the umbilical cord and the placenta also is a rich source of stem cells. So generally we would look for these stem cell properties. They are like plastic adherent, they express some markers. And again, they are also able to differentiate. These are not really important to understand so much, but these are basic tests to understand that something is an adult mesenchymal stem cells or a mesenchymal stem cell. So these are like all the different kind of tissues from where you can get the stem cells, and you can look for the expression of certain markers. Like for anything, you have to do a test. Even when if we are not feeling well, we go to a doctor, the doctor will write down many tests. And then we will see whether those tests we are positive or negative for it. So similarly for these also, these are the markers which you test to understand the stem cells that you have got are truly mesenchymal stem cells or not. You would be studying for this different kind of markers. What
is mesenchymal?
What is mesenchymal? So the mesenchymal is basically something which is derived from the mesoderm. So during development there is ectoderm, mesoderm, endoderm. So example, the mesoderm is something from where we get the fat tissue, the cartilage, our knees, etc., the cartilage, fat tissue, and then the bone tissue. So these all arise from the mesoderm and the mesenchymal. They are mesenchymal by origin. So these are some of the advantages of the mesenchymal stem cells. It's easy to get. Unlike the embryo, which is very hard to get, this is very easy to get. They can also divide and make more cells. The cells you can keep and they will give rise to more cells. Then they can give rise to something like I was just saying, the fat cells, bone cells, cartilage cells. They can also migrate, means the cells can move. Inside the body, they have a migratory capacity. They can move from one region to the diseased region when they are transplanted. And they are hypoimmunogenic cell. That is one very good thing about them, which means they are not rejected. So if you take these cells, put into a patient, say if somebody's umbilical cord cells is given unto another patient, the cells will not get rejected. So there is no immune rejection there. No associated ethical issues like the embryonic stem cell. So it doesn't form any tumors, etc. And they have paracrine mediated effect. So basically in the body, the mesenchymal stem cell is sitting and resting. When there is a damage, this cell gets active and starts to divide, make more cells, and it will give rise to, it will treat the person's diseased tissue. So wherever you have a disease, that gets treated. And they have also an immunomodulatory function. That means when the immune system, we all understand our immunity. We talk about immunity, that we are weak immunologically, or we have to make our immune system strong. Especially after COVID, we have talked about a lot about the immune system, etc. So with respect to that, they are immunomodulatory, that in case there is an inflammation. So many times we get, even if we get a bee stings us, the whole thing becomes red and it becomes swollen. So that is an inflammation that happens. So these cells have a property as an anti-inflammatory. It can stop the inflammation. So in fact, these cells, these cells have recently also been used in the Covid19 trials. So this is of interest because if you remember the initial phases of Covid-19, when the whole world was under the not able to get a grip over the disease, people did not understand much about the virus. People did not know about the vaccine, or vaccine development had not started. That is the time a clinical trial was started in China with 7 patients for Covid-19. With the only aim that because these cells, mesenchymal stem cells, are known to have a anti-inflammatory property, and in COVID, if you remember, that is the time we also understood it was the lung which was getting very badly inflamed. People were dying mostly because of the lung failure. the lung was getting inflammation. There was a lot of inflammation in the lungs. There was something a little bit more scientific term which was called cytokine storm inside the body. So basically what happened is a new virus entered. Generally, if it's a regular virus, even if we get a cold, the virus enters, our body has an immune reaction, and after some days we become fine because the immune builds up and the immune cells are able to kill off or clear off the virus and it is all fine. In case of COVID-19, what was happening is as the virus was entering, it is a new virus, body was getting certain, especially most of the patients, it was overwhelming response. The whole virus, our own body was reacting like a very overwhelming response it was giving. A cytokine storm was building up. Huge amount of inflammation. Lung was failing as a result of that. Heart was getting eventually affected, kidney next, etc. And it was a complete body organ failure. This is what happened in the initial stages. First, if you remember, within the first 1 year, the phase 1, phase 2, whatever we talked about that time. And so that is the time this clinical trial was started in China with 7 patients. Stem cells, mesenchymal stem cells were put into the patient's body. What they found is at least those patients did not die. Neither did those patients enter the ICU. So they did not have to be taken to the ICU, and that was good enough. This was a time when in Italy, Spain, if you remember, the ICUs were full, patients were lying on the street, stadium, hospital, hotels, everywhere, and people were just dying. So that is the time when this trial was very hopeful because people thought that at least if mesenchymal stem cells are put, people will not have to enter the ICU, more and more ICUs would become available, and people will kind of be not go into a very dangerous state or will not be fatal. They will not die. So this is another of the uses of the mesenchymal stem cells. In general, this shows the clinical trial status which have been all carried out by mesenchymal stem cells. For different tissues, different diseases, it has been tested and the cells have been found to be, when we do a clinical trial, it is phase 1, phase 2, phase 3. Phase 3 is the final clinical trial after which you can put it out into the mass and general population can get whatever it is, like we heard about it even from the COVID vaccine, that phase 3 trials it has cleared, Covaxin, Covishield, etc. We have heard about all that. So that is what a phase 3 trial is. So this is typically when you are growing cells in these rooms are called clean room. Clean room is a room where you grow the cells for when it is going to be put into patients. So these are grown in a very clean environment. Clean air blowing inside the rooms. A person has to be dressed like that. This is much before COVID. So this is a normal way of dressing with or this is not a COVID related dressing. So after COVID we see this kind of a dressing we have seen many places. This is a regular lab person working with the stem cells. And with a mask, cap, there is a full gowning. These are all sterilized gowning. Then these are this huge flask. If you see, this is huge amount of cells being grown. These are called cell stacks because you need huge amount of cells when you're going to put into patients for whatever treatment. And then you can also play with the cells to make them even function better. It's like any other medicine. You will see if for a headache, you might be taking a regular paracetamol. The next one could be a paracetamol with caffeine, which would help in even stronger headaches. Because what the medicine has been improved. Similarly, in the stem cells also, you can make different kind of modifications to it. So you can try to make priming the cells, expose them to certain conditions so the cells function even better. Or you do some genetic modification, you make certain 3-dimensional matrices, you grow in them. So here is where even the role of the chemistry and the physics people come in. So like we always have interdisciplinary research areas where the chemist can tell us what sort of scaffold to use. The physics people can do the modeling to say what sort of a conformation it should all have so that inside the body it can make like a real tissue or an organ. So these are all like different kind of latest research in this field which takes the stem cells even one step ahead and make them better. So in my lab we work with umbilical cord. So this is a real picture. So this is a kind of an umbilical cord looks like when we cut the cord and get it to the lab. So then what we do, this is how the cord comes in, we wash it, cut it into smaller pieces like this with a blade cut to smaller pieces. These are 2 to 3 centimeter pieces. Put them in a tube with saline containing antibiotics. the way we take antibiotic when we have a bacteria or virus. So same way we add some antibiotic to this tube so that because this tissue is coming from another human body, and that's why we have to make sure that it is not contaminated with anything. Then you take this kind of a cord piece, you cut it longitudinally. So you have a cord piece, it's like a tube, you cut it longitudinally, open it up, you would find an artery. There are 2 arteries and 1 vein inside. So there, if you see, it's not possible to see here. There are 2 arteries and 1 vein in a cross section. Once you cut it open, remove the artery, underneath the tissue is the Wharton's jelly. That is called Wharton's jelly. Again, it's a medical term or a scientifically medical term. You cut it into small pieces and we put it in a dish like this. We add some medium. Medium means like food for the cells. Growth medium you have to add. And then we put it in the incubator, which is maintained at nice 37 degree, very clean, right kind of humidity, and the pH, etc. is maintained. And these are small, small, tiny things that you see, which are cells which have come out. These are what the stem cells look like, the mesenchymal stem cells. And this is from a human tissue. So this is a cord piece of tissue. So this is how the cells come. Then we do some test to check whether the cells are really mesenchymal stem cells or not. So we look at the expression of these. So because they can make fat tissue, fat cells, they can make bone tissue. So this is actually fat drops. These are oil drops. In the culture room they can make. So this tiny red drops is an oil redo staining, which would stain oil drops. That means the cells can make oil, fat, it can make. So these are very basic. This is routinely we have to do before we do any other study. So other than that, there is one protein in my group which we work with, which is called Vitronectin. And this protein, basically what we try to understand is inside the body when the cells are put, they see a condition which is very different. You have lot of, inside the cells you have like lot of, in a human body, you would have lot of kind of stress condition. There could be, patient could be having fever, etc. Or there could be like no blood supply reaching. So one of our study we do, try to study these cells and see under a high temperature, fever-like temperature, how do these cells get affected. So here these are like this just to show, none of these you have to understand. This is just to show like different kind of studies which are done to check whether, by which we came to the understanding that vitronectin is one protein which helps in the stem cells surviving under high temperature. And here is like an example. This is a cells look like with staining. So this is a fluorescent stain. It is a fluorescent microscope under which we see these. And you see like some sort of, this is, the red ones is a mesenchymal stem cell marker. The green one is a vitronectin protein that we are studying. So here are certain similar studies to show whether the cells are undergo cell cycle arrest, whether the cells are dying, etc. These are different kind of studies which are done in the lab to understand this. And so finally, these days we are also looking at, currently in our lab, so you know also, you might know about the placenta. So when a baby is born, the umbilical cord is cut and thrown in the dustbin because it's of no use. That is the connection between the cord and between the baby and the mother. From the maternal side also there is a placenta. So that is a placental tissue. So this, we are these days isolating stem cells from there. Again we wash it and we take out, finally we get cells from here. This is a placental tissue. These are the cells coming out from there. These are again certain tests which are being done. So I think, yeah, with that, I'll end the talk. And again, thank you so much. These are some of my institutions which I've been associated with, which has helped to shape me or shape my career maybe. So this is my PhD institute, National Institute of Immunology in Delhi. So this is what taught me to have the patience for research, to think independently, and to be able to hold on to that hope that things work. You just have to keep on working day in and day out and it would work out finally. This is in the US, this is a National Institute of Health. This is the largest clinical center in the world. This is the building 10. It is a hugely big building, the largest building also at NIH. This is Stempeutics. Before joining IISER Kolkata, I was in Bangalore with the Manipal group. This is a company under the Manipal group which is called the Stempeutics, which again works on stem cells. So I came back from the US and I joined here. And finally, this is where I am right now. So I joined here as an assistant professor. Today I'm an associate professor here. And with PhD students, a regular lab, a huge amount of teaching, a huge amount of administrative activities, and a lot of other responsibilities where science tends to take a backseat, but then the enthusiasm still remains. So that's how it is. So thank you so very much again.
Thank you very much for this talk. Looks like that we may not need any medicine or something in the future. If it succeeds, then we can cure everything in the body, whatever goes wrong. But that's the directions that they're trying to go to. Stem cell is a wonderful thing, but relatively new. A lot of other things have to be done. So, but you heard the talks. It's a very beautiful technical talk, more technical normally than some of you. But it's very, very nice talk. Thank you very much for talk. Yeah.
Thank you so much. I truly apologize that I couldn't speak in Bangla in between because because I've grown up in Delhi, so I've not had any formal proper education in Bengali, so I get very conscious. I won't be able to speak science part. Like I'm fine speaking at home in Bangla. So I do, of course, speak enough Bangla at home, but I unfortunately...
That's okay, that's okay.
I go frozen.
Now it's open to questions because she's one speaker at least who finished before the time ended. So that...
Our medical centers over here, they make use of this stem cell therapy.
Here means like...
I move to my question. I'd like to say what Maharaj is showing. It's basically the component of Moringa oleifera. It's drumsticks or shojne, what we say in Bengali. And it has been patented by Dr. Professor Karen Vieira of New Brunswick University of USA.
Okay.
And it's a stem cell product derived from the parenchymal and the mesenchymal cells of the plant products. And they have been implemented together to make this product for immunogenic responses in the body. This one. It's of Moringa Organics, New Brunswick University, Dr. Karen Vieira. This is my observation on this product. And what my question is, the first one is that how far effective the stem cells are in the case of autoimmune disease, where autophagy happens and cytokine storm reaction is one major concern because it's a self-induced process in the body where the immune system does not work properly and it itself engulfs the normal cells of the body. This is my first question. And the second question is that in case of immortal human cell line, like the Henrietta Lacks cells or the HeLa cells, they do have genetic shift after all eventually in all these 50 years or somewhat like that. And even in the cases of monoclonal antibodies, how the analogous activities occur like in the stem cells? Are they analogous? The activity of the monoclonal antibodies or the immortal cell lines of the humankind?
No, so, okay, I think your first and second question, or you're regarding your monoclonal antibody, you're trying to relate in the sense, you're trying to ask, like, how do these cells, how are they, whether they can work in a inflammatory...
Are they analogous in that case for the monoclonal antibodies on the human immortal cell lines? Because they are at the
Not really. So there's a different mechanism of action here. So the thing is, the monoclonal antibodies are very specific antibodies which are against one particular epitope or an antigen. Okay, so it's against, that is what is a monoclonal versus a polyclonal, which is made against multiple epitopes maybe of a particular, whatever, we are talking of bacteria, virus, whatever. In this case, and that is how it goes and neutralizes the action of the antigen. That is the action of a monoclonal antibody. This has a different role. This is definitely anti-inflammatory, and this works the best for autoimmune diseases. I didn't want to go through too much of detail about the clinical trials, but if you see most of the diseases of the clinical trials, Crohn's disease, diabetes type 1, etc., many of the diseases are there, which are mostly autoimmune diseases, and it has been found to be advantageous there, because these cells suppress the immune system, the mesenchymal stem cells. They stop the proliferation of the T cells, B cells. The natural killer cells are, these are the ones which are kind of brought down. The regulatory T cells, which are anti-inflammatory, those population increase. So these mesenchymal stem cells basically directly work on the different immune cells to bring down the inflammation.
What for the cases of multiple sclerosis, the normal
multiple sclerosis also, yes.
And also GBS? Guillain-Barré syndrome.
Yes. Then GVHD also, the graft versus host disease. So when a new tissue is put into a host and it would trying to get rejected, if you put the mesenchymal stem cells, it would try to kind of suppress the immune action.
But that is autophagy system, isn't it? The body is not responding properly to the immune reaction, and the immune system is itself trying to harm the body by engulfing or destroying the normal cells. So in that case, how effective it would be for this self-induced system within the body?
So with respect to autophagy alone, if you're asking whether the MSCs can help in autophagy, so that I am not very sure how much data is available. But all that I can say is, like for instance, even in case of COVID-19, when there is a cytokine storm and the body is affecting itself, when the tissues are starting to get damaged by the body's own hyperactive immune response, that is the time the MSC transplantation can calm down the immune system. It can have an anti-inflammatory role and calm it down.
Thank you, madam.
Yeah.
Good evening.
Yes.
One of my friend has multiple myeloma, one kind of blood cancer, and she has undergone the bone marrow transplantation. So in this case, how the stem cells work in her body?
I believe in the multiple myeloma with the bone marrow transplant, so that is what happens in CMC Vellore. It's very well known for doing this hematopoietic stem cell transplantation. So this is, I didn't get into all that because I thought that would make it very technical talk. So other than the mesenchymal stem cells, another form of the adult stem cell is the hematopoietic stem cell, which is in the blood cells, the immune cells that you have, which is another kind of stem cell, which are express CD34, CD45, like that. So with a bone marrow transplantation, you are using the hematopoietic stem cells most of the time, not really the mesenchymal stem cells. So that would help new blood cells to be made and then would improve her condition, like in the case of a multiple myeloma, hopefully. Because with respect to the mesenchymal stem cells, there's just out of curiosity, I mean out of just general, outside your topic, I'm just saying outside your question. With respect to the cancer treatment, there is contradictory reports. Because on one hand, there are reports which say the MSCs can shrink a cancerous growth. On the other hand, there is also reports to say, because these cells provide a very conducive environment, they prevent apoptosis, and then the cells have an anti-apoptosis effect and all that. So it's possible that they could also promote, they have a very strong angiogenic effect. They make new blood cells. So it could also have a promote cancer. So there are not controversial, I would say, there are both kinds of reports. So it's not yet, we don't know yet. But this is what you're talking about is with hematopoietic stem cell transplantation.
Good evening, Professor. It was really good to hear from you. So I have a question. I seem to know that in case of allogenic transplantation, the hematopoietic stem cells that are derived from umbilical cord, they have a greater HLA disparity compared to the HSCs derived from bone marrow or peripheral blood. So I would like to know what is the reason behind the greater HLA difference tolerance when we use umbilical cord stem cells instead of the bone marrow or the peripheral blood stem cells.
You are saying the umbilical cord derived stem cells or the MSCs, they show
HSC, umbilical cord derived HSC.
Umbilical cord itself will not have the HSC, it would be the umbilical cord blood.
Yes.
So the umbilical cord blood HSCs, you are saying, with respect to the HLA typing, it has more disparity as compared to peripheral blood, and
it has a greater flexibility compared to the HSCs that have been isolated from peripheral blood or bone marrow. Like while transplantation, we generally go for HLA type.
See, okay. So because I don't work on the hematopoietic stem cells, because this involves the hematopoietic stem cell, because you're talking from the blood source. But all that I can say is that with respect to the umbilical cord, you have to remember it's a fetal source. It's an earlier source than your bone marrow or a peripheral blood.
Yes.
But bone marrow, peripheral blood, this come from fully formed adults. This is coming from the neonatal stage or like from a fetal stage, the umbilical cord. So the umbilical cord is considered a much younger source of stem cells as compared to those fully adult grown. So they, as a result, would have much more flexibility, and they would be much better. Even that is what your question is. Of course, the expertise, what you're asking is of a different expertise area. But even in our case, we find these cells are much more, what should I say, plastic. The term used is plastic. That is, they can be modulated much better than the fully formed adult cells. The reason being, this is a very young source.
Okay.
Because it's coming from the baby, from the fetus, from the fetal origin.
All right, thank you so much.
So I assume there's the same reason, yeah. Yes.
Good evening, Professor.
Good evening.
I have a question. Could you please explain how stem cells are used in cosmetic industry?
Okay, so that's a good question. In the cosmetic industry, what happens is, it's actually like real this thing. So more than cosmetic, it is also used in the burnt wards also. So it has promise there also, because if you, as the cells are growing, this mesenchymal stem cells, they secrete lot of factors. Okay, secrete means a lot of factors, proteins, very good growth factors, proteins are coming out from these cells. So those, if you take the conditioned medium, the liquid part of that, what is coming out of the stem cells, if you take that liquid part, it has anti-scarring and healing properties. So if somebody has a scar on the face or anywhere else, a burnt patient, from a burnt ward also, like say if you see a lot of people get huge scars, if you apply that, it is thought to be having a wound healing property and also healing scars and having a regenerative effect. The cells are known to have a regenerative effect, that's how my title was also. So as a result of that, any kind of a skin, you might have patches and you might have whatever problems with your skin. So what in the US I've seen cosmetic products which claims conditioned medium from stem cells, and you would have flawless skin. I don't know how much of the flawless skin, but the promise is there, because this could have regenerative factors in them and anti-scarring properties. So any scar and all that should go away.
Thank you.
Any more questions? Okay. And that was again, we thank the speaker, all of us, with an applaud. Thank you. Thank you very much.